gonna learn about the systematic review, which is it is considered as a tip of a pyramid. Okay, it is like a high-end research. Whatever the article comes from a systematic review is like a high-end research. Okay, so you in this session you're gonna study what is systematic review, how the systematic really works, and why we are conducting the systematic review. So there is a brief introduction I will do, and I will say the process of the systematic review. Okay, so the learning objectives, like I mentioned, what is systematic review? We will see the small difference between systematic review and traditional review or a narrative review. And we will see why the systematic reviews are important and the steps for performing a systematic review. Okay. So this is a graphical illustration of a Cochrane given by the Cochrane. Okay. So Cochrane is like a group of journals like the Medline or PubMed, like it is a group of journals. Okay. So they gave this illustrations. Okay, how the systematic review really works. Okay, so according to them, it's like a filtering. So you filter each layer of review, the literature, because see, when you go to any sites, you find a bundle of articles, right? So you filter it in each layer and you produce a good quality data. So this is one graphical illustration. So we will see each step of this filtration, how the articles get filtered in each step, and a systematic review is formed. Okay, so this I want you to see this hierarchy of the of the scientific evidence. See how you can see. So you can see the strongest to the weakest, and when it comes to the strongest, you can see meta-analysis and systematic review. It is at the top. Like I mentioned, it is at the top of the pyramid. Okay, so as you can see the case report, which is at the very bottom, and then comes the animal trials, the cross-sectional studies, case control studies, cohort studies, RCTs, and then the tip is nothing but the meta-analysis and systematic review. Okay, so what the systematic review is. Okay, so it is basically a transparent procedure to find, evaluate, and synthesize the results of independent studies. So what happens is there will be you can see those diagrammatic representation. There will be a lot of individual studies. Okay, so it is a kind of a procedure where you find what are the similarities between these individual studies, you evaluate them, and you synthesize the data that is required for the clinical purpose, like for a patient care. Everything is done because at the end of the day, if medical sectors, everything is for the patient care, right? Okay, so you have two kinds of drugs. Now you have to find which drug is good for a patient. So what happens? They do RCTs. There are a lot of RCTs happening. So even in the RCTs, the observation might be different. Okay, so systematic review is something you compile everything, you filter, you find, and then you analyze, and you say, okay, it is a place where you find the result of all the studies. Okay, so the procedures are explicitly defined in advance, okay, to ensure that the process is transparent and can be replicated. This process is also designed to minimize the bias. So there will be no bias in systematic review. Okay, the unique characteristics of systematic reviews. In systematic review, the inclusion and exclusion criteria should be very clear. Okay, so what kind of studies are you going to take? Are you going to take RCTs, or are you going to take an observational study? You might be, or a cohort studies. You should be clear about the inclusion and exclusion criteria. You should be very explicit about the searching strategies, where you will search the articles from. Do you take it from a PubMed or a Medline, or you might just take from a Medline, or you might take from Medline or Cochrane, or you might take from Embase. So you should be very explicit about the searching strategies. Okay, systematic coding and analysis of the included studies. You have to code them specifically, like you do a data entry, for a patients. If you take okay, this is a patients have come, so this is a data, this is the HB level. Or if you take, okay, this persons have taken drug, yes, 1 I give for a person who has taken a drug, and 0 for a person have taken a placebo. So the coding should be very accurate, okay, because that is what decides your results, right? And analysis of the included studies, then meta-analysis come to place where possible. So if your data are good, if you get a proper amount, like 3 to 4 studies, then you go for a meta-analysis where you compile the data and give the results, and you tell, okay, this is the outcome of the study. So let's see what this meta-analysis is. It's an optional component of a systematic review. So here the statistical analysis of the results, okay, from all the individual studies. So you got some results, right? So you enter some results. So of these results, you analyze, okay. So what it does is basically it increases, okay, it increases the power in the sense, it increases the quality of the research. Whatever your research doing, it increases its quality, and it improves the estimate of the size of the effect, okay. And it resolves the uncertainty, okay, like I mentioned, okay, this drug or this drug. So it will help to resolve that problem. It brings together the best available results. Means when you do an analysis of the analyzed data, you will get a better format or better results, okay. So it is like bringing together the best available results, okay. Next, let's see what is the difference between a traditional and a systematic review. Why we do conduct a systematic review? Because there is something called a traditional review or a narrative review, right? So why are we going to do a systematic review? What is it? Okay, so what is a traditional review? Okay, in a simple words, if I have to say, like, see the author, okay, if you are an author, you pick a bunch of paper. If you're going to write a narrative review, you just take a set of papers, okay. Now you want to write a narrative review on a particular topic, okay. So I want to see any kind of treatments in for example, you have to see whether antidepressants causes any risk of increases the risk of suicide, okay. Now what you will do, okay, you will take up studies which will correlate with this title, okay, or with this what are the aim of your studies. So basically antidepressants with the whether they can have a risk of suicide among the patients, okay. So now you will take a bunch of studies which will go with this your aim or your title, right? So what happens, you just take a random studies which you get in a first place, okay. When you do a search in a PubMed or a Medline and Embase, Scopus, Web of Science, you just do some searching and you find some of the studies, and then what you do is just start writing a paper based on that. So what happens on this, there will be bias, because you might have left some studies which might have shown a negative effect, okay, or there might be study which might be saying a very contrast results. So whenever you conclude your paper ends, okay, there might not be any effect of antidepressants among with the suicide, with the risk of suicide. There might be really happening some risk, okay, which you might be completely neglected because of the bias, because you're randomly selecting 10 or 15 article and you're just blindly writing. So there is a complete biasness, okay. So narrative review will be full of bias, and the results what or the paper comes out, it will not be that of great quality, okay. So like see, like here it is mentioned, the searching, the quality appraisal, the data synthesis are often not documented. You just start writing, it will not be documented, okay. Next, since the scientific methods are not routinely used to identify, because you don't undergo a procedure method or a step-by-step method, so it cannot be verified, okay. Greater risk of author and selection bias. I told you, just select whatever articles you get, you don't go a step-by-step process. So comparison of traditional and systematic review. So this traditional review will be subjective, okay. So it will be based on the subject. So what subject you're gonna change, you're gonna study, okay. Maybe the effect of the diuretic therapies in the in new AKI. So you just see this subject, okay, AKI is there and this kind of a drug is there. So what, let's see what is we are going to write, or what is the outcome, whether it is a good means it really causes AKI, or if it causes AKI, what is the pathophysiology, what is going to happen, what are the, you will list out some of the diuretics. So this is what it is happening. So there will be just subjective. In while in systematic review, there will be objective. Objective means you will clearly mention what is the objective, which I'm going to tell you in a coming slides. Okay, I don't want to explain right now. So let's see what are the differences right now. So the traditional will be again the broad questions. The broad, it will be like you will tell treatments for encephalopathy. Okay, so all the treatments. So it will be producing a mass of data, and you just it will not be very specific. However, in systematic will be a narrow questions. Okay, this particular drug in the treatment of this particular disease. Okay, there will not be there like all the treatment for chronic kidney disease. No, it will not be there in that way. Okay, next, methods will be unclear. There will be, you will not follow any methods, right? You just go on writing because it is a narrative review. However, in systematic review, it will be very clear. The studies are appraised. Okay. So like I mentioned, this is the quality of evidence. So observational studies, you can see the experimental studies and the critical appraisals, that is meta-analysis and systematic review. This important, the systematic review and meta-analysis is. Okay, so why we are conducting systematic review and meta-analysis? Because there is a overload of informations. So like you can see, it is like you arrange. Okay, you have lot of data, you have lot of papers, you arrange into a single sheet. Okay, if you consider, you take the data from various journal and then you arrange. Okay, so some of the guidelines. Okay, healthcare decision should be informed by the best available research evidence. Okay, so many guidelines, like for example, okay, for this treatment, okay, the treatment of cardiovascular disease. So any guidelines for the treatment or anything should be that comes because of the research evidence, right? So this research evidence, I told, should be very accurate, because that decides the patient care. Okay, in the healthcare sectors. So this should be very precise. So many guidelines, it will rely on the previously published systematic reviews. Okay, practice guidelines, the quality depends on the rigorous systematic review methods and high quality primary studies, like RCTs, systematic reviews, evidence-based practice, and clinical practice guidelines. So this is why it is very crucial that whatever the systematic review anybody conducts, it should be very rigorous and it should be high quality. Systematic review is itself is a high quality, it's a research, high quality research. Okay, next, known and unknown areas of study. Systematic reviews can show which treatment and prevention methods have been proven to work and what remains unknown. Okay, because you compile lot of data, from the various studies. Okay, systematic reviews are important for pointing to areas where more research is needed. Okay, for example, if you consider, okay, in the case of like Indian sectors, if you see how the quality of life in different patients. Okay, if you consider a liver cirrhosis patients, what is the quality of life? Now we know, okay, the quality of life, in countries like India, we don't focus on quality of life. So we can say, okay, the quality of life, we are not focusing on this part. So if we are we can say, okay, this can be done to focus on the quality of life of a person. So we can develop a various strategies, maybe in giving them some of the patient information study by forming a team that helps the patients to understand that it's not just a treatment, even the quality of the life is important, okay, teaching them or conducting them some kind of a community programs. So we will understand some of the reasons, some of the areas for where more research as well as more outcome, whatever, like what solutions to be come. For example, if you're going to see a systematic review of various biomarkers, so you conduct a biomarker based studies, for example, for finding a one particular, okay, for example, kidney. In a kidney, to see some kind of a kidney injury, you give different biomarkers and then you do a systematic review. And in the systematic review, you will understand that none of these biomarkers, like which particularly in the focus, have having a great effect, means they are not fulfilling the criteria, or they are really not finding the whether the disease is present in an early stage or not. So then you will tell, okay, these biomarkers are not very efficient, so there should be a vigorous research to be done to find a biomarkers to diagnose this particular disease. Okay, this is how the systematic review works. Okay, systematic reviews are the basis for what is called as a evidence-based medicine or healthcare. Okay, so I want you guys to see the steps. Okay, these are the steps to be followed to conduct a systematic review. First one, develop a focused research questions. Okay, first, the primary step is to develop a focused research questions. The second one is to define the inclusion and exclusion criteria. So once that is done, we have to search the literature. We have to select the studies. Once the search of the literature is done, we have to select studies. What are the studies are required? Next, we have to assess the study quality. Okay, so once the assessment is done, we extract the data. So what data are required for us? Okay, next we analyze the result, or present the results, or interpret the results. Okay, and draw a conclusion. Then update the necessary. So once your systematic review is done, so you can do further systematic review even after 1 or 2 years, because there will be some other centers who is doing this. There will be constant study or research is happening, right? If you conduct RCTs of this particular drug, so you like, for example, your systematic review will be till 2007. Might happen in 2007. So after that also there are a lot of research is happening. So after that you can start from 2007 to 2010. So that way you have to update as necessary. So let's see now. I have you can see the first point is highlighted. So to develop a focused research questions, let's see how to develop a focused research questions. Okay, so research questions. So whatever questions we are asking, okay, or we are trying to answer, it should be clearly defined. Okay, it should be focused systematic review, because it should be focused, like what you're doing. You can't just say treatment for all cardiovascular disease. No, it is not focused. Okay, it is not very clear. If you see, it is not a good questions. It is not a good research questions, because your questions should not be broad. It is a narrative review. It should be very narrow. Okay. For examples, I told you, you can't say that all the treatments for a cardiovascular disease, but instead you can see, NSAIDs for cardiovascular disease. Okay, so you have to take a particular drug. Okay, one group of drugs for the treatment of cardiovascular disease. So this is how it works. Means you have to define it clearly and it should be very narrow. Next, the research question guides, it guides the authors in working through many stages of the systematic review process. Like in that, you have to defining inclusion and exclusion criteria, searching the literature, because search literature will be based on the question you formed, right? So it is when you search, you will search cardiovascular disease, you will search NSAIDs, right? Selecting the studies, extracting the data. So every step depends upon your research question. So research question should be very clear. Only then you can go with all these steps. Okay. Next, So when you form a research question, so as an author, okay, so now you might be working in a particular field, okay, maybe your area of the interest, maybe you're working on a kind of kidney diseases, okay. So if that is the cases, you have to see your question, you developed your question, okay, then you have to see the final criteria, okay. Research question should go final criteria, how feasibility, whether I can really do this study, okay, whether it is feasible enough, whether it is interesting, whether it is novel. Interesting means it should be having some result, the result should be useful for the patient care or in the healthcare system. The novelty, whether, see, if somebody else also he is doing, then what is the point of you doing that? So there should be novelty in the systematic review. If some person is doing a systematic review, then you cannot do the systematic review on the same topic. Ethical, whether, because sometimes, ethically also sometimes, whether you can do, okay. And the relevant, whether you can't take something else and something else, it should be relevant to the subjects. This is very important, the PICO frame. So once your question is framed, okay, this is my questions, okay, then you have to see the participants. So for example, so particular kind of drug in a cardiovascular disease. Now what kind of participants you are seeing? You are seeing a patients with the cardiovascular disease or in a cardiovascular coronary artery disease, okay. Your participant should be very clear what kind of participants you're gonna say, okay. If you're gonna say, if you're gonna see, for example, if, okay, some kind of therapy, okay, immunotherapy in this kind of cancer patients. So which cancer patients? Okay, for example, hormonal therapy in breast cancer patients. So your participant will be a patient with breast cancer. Next, again, patients may, what is the age group, what is the, what is, you have to define the patients properly, you have to define the population very accurately. Next, intervention. So intervention is what? Immunotherapy. So what exactly, what kind of no, I'm sorry, I was talking about hormonal therapy. Yes, what kind of a hormonal therapy are you gonna give? So the intervention should be precise. Next, comparison. You are gonna compare this hormonal therapy with the patients who have received or not, right? Who have received hormonal therapy and who have not received hormone. So there should be a comparison group and the outcome. So what is your outcome? So what outcome you want to see? Whether it is really effective in the treatment of the patients or whether it will produce some adverse effect. So you will see the outcomes of the therapy. So this is how you do. So you frame a questions and then you list the PICO framework, that is patient, intervention, comparison, outcome. This is a basics. This is a basics of the systematic review. So this is still, we are in the foundation of the systematic review. So you have to define a question and then you have to do a PICO framework. Next, define the inclusion and exclusion criteria. Like I mentioned, you have to be very accurate when you do a inclusion and exclusion criteria, because this will tell how many studies will fit into your systematic review. Okay, this will tell, what your studies will be, how your data is gonna, come. So one of the features that distinguish a systematic review from the traditional review is pre-specification of inclusion and exclusion criteria. In narrative, you don't do this inclusion and exclusion criteria, but in systematic, you will do inclusion and exclusion criteria. Okay. So the inclusion and exclusion criteria, it will be, again, I told, based on the PICO, your population, intervention, like hormonal therapy also, you can see which kind of hormone therapy you are gonna give or which kind of therapy you're not gonna give. So again, very specificity. It's very specific. Again, comparison group, what kind of comparison? Again, outcome. So it might be you're seeing the good outcome or it might be you're seeing the bad outcome. So again, the good outcome is you can choose only good outcome. So your inclusion will be this. Okay, this is how the inclusion and exclusion works. Next, study types. Either you have to very you should be very specific that, okay, in this systematic review, I am going to take only randomized control trials. So then you can't take other kind of study. You can't take cohort studies. You can't take observational studies. You can't take retrospective studies. Okay, you should be very specific. Okay, I am going to take this kind of studies. So then your study will stick to RCTs. Okay. Oh, no, in my study, I want to see whether this biomarker is working on this population. So I have to see the observational studies. So then I have to only go for the observational study. So this will again limits, like, so what happened when you do a literature, literature search based on your research question, you might find thousands of papers. So once you put your inclusion and exclusion criteria, the thousands will become a few thousand, right? So it might be 52,000, 48,000. To that, it might come up to 12,000 or 8,000 based on your inclusion and exclusion criteria. Okay. Next, search the literature. So searching the literatures, okay. So this is, again, I told it was based on the inclusion criteria. Okay. So we have to search comprehensively based on the basically two things, your terminologies and the databases. Okay. What terms you are going to take. So basically the research question based on the PICO. So what group? This is a group. What is your intervention? This is your intervention. So this terminology should be very specific. What database you are going to use, where you will conduct your literature search, whether it is Embase, whether it is Cochrane, whether it is Medline, you should be very specific. You have to mention, we conducted a database search in this. Okay, this sites. Okay, this is search engines. Okay, search for gray literatures. Okay, here means not commercially published papers. Search for unpublished studies to reduce the risk of publication bias, because systematic review, I mentioned, it should completely cover all the studies that happened with related to your research questions during that span of time. So if you say I conducted a systematic review from this year to this year, you have to completely, you have to cover all the studies, and you should never miss a single study. So recommended databases, like I mentioned, PubMed, Embase, Cochrane. So these are the common database search that you're going to do when you do a systematic review. Next, supplementary databases. So interdisciplinary databases, Scopus, Web of Science, specialized databases, Sinhal, Pedro, PsycInfo. So these are some of the other specialized databases that are used. So you can search your, studies in this databases as well. Next, search strategies. To identify maximum number of studies relevant to the search questions, so the search strategy should be systematic, transparent, and reproducible. Okay, the database specific control vocabulary terms. Okay, for example, if you're gonna write, okay, chronic kidney disease, you have to write chronic kidney disease or CKD. So it should match the vocabulary terms, all the text words. Okay, should include in the search strategies. Next, supplementary searching techniques. Okay, like Scopus, Web of Science. Next, you can also see the some of the selected journals, conference proceedings. Sometimes what happens, sometimes some data will be published in some conferences, so you will find results. Okay, like for example, if it's an observational study, so they will just give in a conferences, so the data from that as well can be selected. Okay, or what happens, you can ask some of the experts to give. Okay, since you do a search, you will find a particular person will be there in a many kind of research. You can ask him, okay, so what is there any studies that you're going to do or any results, because we're going to do a systematic review. So that also can be done. Unpublished and gray literature. So here the inclusion of unpublished and gray literature may minimize the potential effects of publication bias. Okay, So gray, what does this gray literature mean? Okay, it refers to the academic, business, government, or industry print or electronic literature that is not controlled by commercial publishers. Like I mentioned, the conference proceedings or the research reports or some government reports or dissertations or theses. Okay, or organization websites. So sometimes what happens if you're working on a particular organization, for example, the Oxford University, it will have its own website and they publish in their own website, which might not be, you might not have access to that. So this is, you have to take access. So this is gray literatures, okay? Or some dissertation. So it will also have some data, right? It might be happen in a small center or a single center with a small sample size. Still, it will make a differences, okay, in the yes. Is there any questions? Okay, so it will also make differences in the your systematic review. One second. Okay, so dissertation, theses, there is a site called Shodh Ganga where you find the different theses, okay? Or you can go this clinical trials, research where they put this reports, all the reports. You can find the various studies that is unpublished. You can take the consent, you can go to the expert, you can mail them and you can ask the studies if they are not published and working on it. Next, so again, the conference proceedings, MBA, Scopus, Web of Science, Google Scholars, all this, okay? Next, again, I told, like I mentioned, unpublished data literatures will be again clinical trials, centralwatch.com, open trials, World Health Organization, clinical trials registry. All this registry will have a certain RCTs that is registered, okay? Next, compile. So compile the, so once you found, okay, you did a literature search and you found thousands of articles based on the search, search tool, what you gave, or the search, words, what you gave, you found, literally a lakhs or a thousands of papers. So now what you do, you take a reference management software, either EndNote or Mendeley, and then you arrange. So what happens, this will help to remove the duplicates. So what you might have searched in the PubMed might be present in the Embase as well, or might be present in the Cochrane. So what happens is, so it will be again what happened, the same data. No? So this will remove the duplicate records. So document search process, okay. So searching process, again, database, date search. Date search means what year to what year you want to search. Search strategies. Search strategy means you are choosing a study which will be conducted only in the English language or only on the human beings. So this is all some of the strategies that to be applied. Limits, again, date ranges, publication types. Publication types means whether it is RCTs, observational studies, language restriction. Like I mentioned, whether it is published in English, if it's other language, no. And if you find a complete data, only then we are going to take these studies. If the just the abstract is present, we are avoiding that kind of studies. So these are some of the search processes. So you have finished the three things. You have searched the studies. You have ample of studies. Now you have to select them for your systematic review. So this selections will be made on the review of title. So when you read the title, you will understand few things. Okay, this is what the study is. Sometimes it will be completely different from your, what you need. Some you can completely ignore that studies at that time. And once the study title is done, the next part, okay, you filtered something. Now you got some studies. Then you have to read the abstract. So if the abstract you feel it will go with your systematic review, only then you have to take them. And then full text studies, okay, with your inclusion criteria. Next, keep a record of reasons for inclusion or exclusion. Okay, why you included this study, why you excluded this studies. So you have to do all of this stuff. Next, again, the tools for sorting will be EndNote, or you can do via Excel. Next, assessment. So this is very important. Okay, assessment of the quality of the study. So following the full text review, assess the selected study for risk of bias and study quality. So tools to do this is Robin. So these are the different tools you can find. So this will help to find the risk of the biasness. Okay. So basically, when anybody does, okay, systematic review, a single individual cannot do a systematic review. So there should be a multiple reviewers. At least 2 reviewers should be there who should do this review independently, okay, without each other's input. Okay, they should be very independent. Next, extract the data. Now you understood, okay, this many papers are there. Now you have to extract the data. So from the selected studies, you have to extract the data, okay, whatever is required. So at least 2 review authors should independently extract the data from the study reports. Next, once the data is formed, what you have to do? You have to analyze and present the results, right? So in narrative synthesis, what happens? You just summarize, explain in the words. But here, sometimes there will be quantitative data also. So it will be statistically analyzed and then it is summarized. So that is nothing but the meta-analysis part. So that is where the meta-analysis comes to the picture. So where you analyze the data and then you statistically analyze the data. You just not analyze the data with the words, which you do in a narrative review. Instead, you do it in a statistical way. Okay. Next, tables and figures, flow diagrams. So you have to do some flow diagrams like PRISMA charts. Next, summary of the finding table, the forest plots. So I will explain each one by one. So what it is. So PRISMA flow diagram. So PRISMA means PRISMA flow diagram is what is. It depicts the flow of the information. How you went from the research questions, literature search, inclusion, exclusion. So how your studies have flown. From the ample of papers, how you came up to this 10 papers that you have taken for the systematic review, or to the 4 papers what have come to a systematic reviews. Okay, it depicts the flow of informations through the different phases of systematic review. The documents the number of studies that remain after each stages of the selection process. Okay, each stage. Okay, it was basically in the initial stage, it was 90,000 papers. Okay, after the first step, it came back to 30,000. So 30,000 to once you put inclusion and exclusion, it came back to 10,000 paper. And then we read the abstract, and then we read the entire paper, and then we just came back to 30 to 40 papers. Again, risk of biasment. There might be some duplicate papers, and we didn't get the data what we wanted for the meta-analysis. All that things, it will finally end up max to max, very few papers, right? If it's good, if it's more, if it is better. So we came to 12 papers where we are going to conduct a systematic review. So this is what for the meta-analysis of this. So you can see here how it goes. See, identification, screening, eligibility, how much we included. Okay, this will finally go to the meta-analysis. You can see records screened, records excluded, assessed, excluded. For quality synthesis, the systematic review, and then the quantity, where the meta-analysis comes to the picture. Next, summary of finding table. So providing the key informations for the quality of the evidence, depicting the magnitude of the effect of the intervention. Next, to illustrate the sum of the available data on all important outcomes. Okay. Summary means what are the, you can see the, when you do HDR or LDR brachytherapy for oral cancers, you can see the outcome. Outcome will be local recurrence, mortality. So you can see here how they have illustrated the findings. You can see the number of patients, the relative effect. Okay, the interval, the confident intervals, all this you can see how it is. See here, Ripin. The grade 3 or 4 complications. What happens? The number of patients, 5. So it is low. So all these you can see based on the various studies. Next is the forest plot. So forest plot is nothing but it is the illustrations. So it is like a graphical representations where you represent your systematic analysis data that is basically in the meta-analysis. Okay, so how you describe. So when you see in a visual, it is a visual representations. Okay, so this is how the forest plot looks like. Okay, so again, here in the forest plot there will be something called as the homogeneous data or the heterogeneous data. Okay, so forest, in the forest plot there are various segments here. So you can just see here this vertical line, it will tell how much studies. Okay, 1 2 3 4 5, all these the results from the individual studies. Okay, so here the horizontal effects of the study, so it will be based on the data analyzed, like mean difference, odds ratio, hazard ratios. Okay, so what happens here is, if this plot it comes towards this vertical line, then it is 0. Okay, so there is no difference of your study. So if this is on the same line and it's towards the same, for example, so if this is the thing, this here, this is not there. So if it's on the same okay, basically it is here, not this side. Okay, so if this is the data, this means it is heterogeneous. Okay, it is not homogeneous, the data is not fine. Maybe you took a observational study and RCTs, maybe you did not define your research question properly, maybe you took a age group of elder people and younger people, so because of which there was huge deviations, so your data did not come out very well. So this is nothing but the heterogeneous and homogeneous thing. Okay, so homogeneous is nothing but here, like this. So it will be present in a same region. Okay, so this is about the forest plot. Okay, so what happens, studies, okay, studies with the scales or the points to the left, okay, towards the left, will have effect sizes that favors the intervention group. So for example, if you a particular drug is there, okay, so drug, if this is towards this side, it means the intervention, means your study is showing a good intervention for a particular statins. Okay, if statins is in the treatment, so it's showing a good. Okay, however, if it's towards the right of the vertical line, then favors towards the placebo or the control. For example, if it's a antiviral drug, so if your data is, you can see here, if it's towards the left, then it is, the data is good. Okay, if it's towards the right, then they are saying favors towards the the placebo or the control group or the comparison group. Okay, next, size of the each square or point indicates the weight of the studies. Okay, so whether the study is good, the outcome of this effects. Okay, the outcome, the weight of the study, and this line, no, it represents the confidence interval. So if the confidence interval of the study overlaps with the vertical line, it means this effect is not statistically significant. Like I mentioned, if it's correlate with this straight line, then it is not statistically significant. So this is about the forest plot. Next, interpret results and draw conclusion. So statement of finding, discussion and conclusion. Information on all important outcomes, including adverse outcomes. Okay, so everything need to be interpreted. Quality of the evidence for each outcome. So like you saw in a oral cancer, so mortality or recurrence of the cancer. So the evidence for each, it should be, the quality of evidence should be produced. How values and preferences may bear on the balance of benefits, harm and cost of intervention. It's based on the intervention, the kind of intervention you're gonna do. Next, update as necessary. So if there is, like I told, the update, okay, the recent studies, or you have done your studies 3 years ago, now you have to update. So update as necessary. So currency of the questions asked, the updating, the maintain credibility, or new evidence, like I mentioned, new researches, new methods will be affect the studies. So this is how you like I started with this same illustration. I hope now you understood how this filter works in each steps. Okay, next, registration tools and archives. So there are Cochrane tools, like I've mentioned some of the tools like Robbins for assessing the bias. So all these tools, and it will help, the handbooks, it will show how the systematic reviews, Cochrane reviews will be there present. Okay, the same. These are the software that is used for the recommendations, assessment, development, and evaluation. Prisma, like I mentioned, how much studies are included, how much studies are excluded. All this, there is something you can just type in the Google Prisma chart generator. You can see to develop a Prisma chart. Next, Prospero is something, it is very important. So before you start, means once you start with the data synthesis, you have to register your study in Prospero. Okay, it is like a international prospective registration. Okay, so what happens? So once it is registered, it's not like anybody else can't do what your study is doing. So it is very important to register your study, the planned study in Prospero before conducting the study. So softwares for systematic review, softwares for like these are some of the softwares for systematic reviewing. You can see some softwares. You can just Google search and you can find some of the softwares that are used for systematic reviewing and even for the meta-analysis part, the statistical things. Okay. So this is about the systematic review and a small highlight on the meta-analysis part, the forest plot. So basically, if I have to say what it is, so I just want to summarize. Okay. So in this session, you studied what systematic review is. We compared systematic review and meta-analysis, and we saw how to analyze the data. So what happens? Like, consider you as an author. Very simplified way, I'm going to say this. See, you are an author. So you have a research questions. Basically, first you have to do is PICO. That is patient, intervention, comparison, and outcome. You have to see what kind of patients you want and what kind of intervention you are going. And this intervention is compared with which group. Okay, you can compare the groups as well. And the outcome, what you're going to get. So once this is done, you will conduct a literature search. Okay, so this literature search will be ample of literatures is present. Embase, Medline, Cochrane. So, or else you can go to the gray literatures. Okay, you can go to the unpublished data. You can go to the theses, clinical trials, registers. You can also ask the expert, direct, you can mail the questions, experts. You can ask the data, if they have some. So the literature, so you framed the questions, you did a PICO, you did a literature survey. Next, pull all the articles, read the abstracts. Read the abstracts, okay, which comes closer to your studies, keep them. All the studies, you keep them. Rest, ignore. Okay, but why you have avoided them? Just put a list of them. Okay, so once that is done, read the complete article. Once abstract is done, read the complete article. Okay, so every step should be done by a multiple reviewer should be done. Okay, so now the full articles is read by the multiple reviewer. So you can see the pictorial representation I have given here, the multiple reviewer reviewing the spreadsheet of the full article. So you have thousands of full articles. Full articles means you don't store it that way. You just read it in the full articles. Okay, so the multiple reviewer. So so one reviewer might feel, okay, this looks like a paper for our review. The other person might not feel that in that way. So what happens if that conflict is there between the two reviewers? Then there should be a third reviewer who will give the decision whether that paper should be there or not. Okay, so this is how the systematic review works. Next, along with this, you have to extract the data. So data extraction, what are the data you need? Okay, in case of a treatment, so what is the treatment, whether the side effects is there, all that things, or whether the demographic details. Okay, all this again, it is conducted by the multiple reviewers. Next, the analysis. The what analysis you are gonna do? The forest plot. You will consider this studies, and you will see the outcome of the studies. So this is about the systematic review. This is how we conduct a systematic review. I hope you guys, understood how the systematic review works. You can ask any kind of questions you have. So I'm ready to answer any kind of questions. Okay, so how does one find unpublished RCTs or studies which are not of Indian origin? So you can see the clinical trials, registers, like I've mentioned some of the clinical trial registers, right? You can see that. Maybe we will share this video recordings. You can see I have mentioned some of the list of the, registers. You can go through that registers and find some of the studies. Yes, anybody if you are from a research background, or if you are working in into an academics, you can start do a systematic review. But I told a single person cannot do a systematic review. Systematic review need to be conducted in a team, okay, which should have two experts, okay, who is guiding you, and you the two reviewers. I told two independent reviewers who should conduct a review, okay, separately, independently, because there should not be bias, and the expertise what you're working on. Any other questions? Yes, you can add unpublished RCTs in the article, okay, in the review, in the systematic review you can add. See, in the systematic review you add in the sense you take extracted data, right? So if you want that kind of data, for example, if the unpublished, okay, it might, okay, the unpublished RCT might be based on the the antiviral drug. So if it's working better for the person, so you can take their data. If you, for example, antiviral, so if you have to see, okay, which, okay, for example, HBV antiviral drug, so what is the HBV load? So all this, whether it really improved after giving this antiviral drug, you can take the data. You have to take the consent from the person, of course, it goes in that way, and then you can include that in your study. Since a systematic review takes a lot of time, so when you do all this process, by that time, no, they might have even be published their data also. So in the next class, you will see how you, about these citing tools, where, okay, where I will tell how to, the reference managing, how to do, like I told, when you insert in the Mendeley or, in the Zotero, there will be duplicate. You can find the duplicate data where you can, avoid that, the duplicate data produced. So that is, you're going to study in your coming classes, and maybe that is tomorrow. I hope there is no other questions. If there, you can just put it on a chat box. So I'll take it as there is no further questions. I think we can wind up with the session. Thank you.
Yes, thank you so much, ma'am, for your lecture, ma'am.
Okay. Thank you so much, everyone, for your time and patience.